Right now, inside your body, your immune system is making, millions of life or death decisions every single second. It's patrolling your bloodstream, your organs, your tissues, and it is basically acting on one fundamental question.
Right, the classic, is this cell me or is it not me?
Exactly. If it's me, it gets a pass. If it's not me, it gets destroyed. But what if I told you the answer to that question is almost always a lie?
Or at the very least, it's a massive biological negotiation.
Yeah, a negotiation. Welcome to this deep dive. Today, we are taking three ancient Greek combining forms. So that's auto meaning self, hetero meaning different, and homo meaning same.
Three very small words doing a lot of heavy lifting.
Seriously. We are going to unpack how these three little prefixes actually act as the ultimate skeleton key to understanding, well, modern biology, immunology, and genetics.
The journey of these three words is wild, honestly, because when you see them on a medical chart or in a textbook, they seem, incredibly tidy, self, different, same. But biological identity is never a fixed, absolute point.
It's strictly relational.
Yes, exactly. It's always relational.
And that is actually the central theme I want you, the listener, to keep in your mind throughout this entire deep dive. Whenever we discuss what is self, same, or different, I want you to ask yourself one question in relation to what?
Which, I should point out, is so much more than just a vocabulary drill. The shifting definitions of these three prefixes have literally dictated how surgeons make clinical transplant decisions.
Life or death stuff.
Exactly. They drove major Nobel Prize-winning rivalries in the lab, and they reshaped the everyday English vocabulary you use without even realizing it.
It's fascinating. So we really need to start at the turn of the 20th century. Biology is undergoing this, massive transformation. Scientists are suddenly peering into invisible processes, specifically genetics and immunity.
Right, and they desperately need new language to describe what they are seeing, because the vocabulary looked tidy on paper, but the actual living science they were trying to pin down was incredibly messy.
So messy. Let's look at genetics first to see how this started, right?
Yeah, so in 1902, there was an English biologist named William Bateson, and he was working alongside Edith Rebecca Saunders. They were grappling with Gregor Mendel's newly rediscovered experiments on key plants.
The famous pea plants.
The very ones. So Mendel had mapped out these specific inheritance ratios, but it posed a profound conceptual problem for biologists at the time.
Because, if a pea plant showed a dominant trait, say a specific flower color, you couldn't just look at it and know why it had that color.
Oh, right, you couldn't see the genes.
Yeah. Was it because it carried two strong hereditary factors, or was it carrying one strong factor that was sort of masking a hidden weak factor?
Exactly. They needed a way to label that invisible internal difference. And Bateson realized that standard English just didn't have words precise enough for this. So he reached for ancient Greek.
Like you do.
Like you do. He coined the term homozygote for an organism carrying two matching copies of a trait, using homos for sime. And he coined heterozygote for an organism carrying mismatched copies, using heteros for different.
It's brilliant in its simplicity, really. It gives you a way to talk about the invisible architecture of life. But while Bateson was doing that for genetics, other scientists were trying to define the boundaries of the self in immunology.
Which brings us to Paul Ehrlich.
Yes, the German physician-scientist working around 1900 with Julius Morgenroth.
Right. Ehrlich was intensely focused on blood serum. He was running experiments trying to understand why blood serum from one animal would rapidly destroy the red blood cells of a totally different species, but, and this was a seemingly unbreakable rule, it would never destroy its own red blood cells.
It just wouldn't happen.
Never. To explain this absolute biological boundary, he coined the phrase horror autotoxicus.
Which literally translates to the horror of self-toxicity, right?
Yes, using that prefix auto, from autos meaning self. Ehrlich firmly believed the body possessed these absolute active safeguards that prevented it from ever manufacturing weapons against its own tissue.
So he thought the system was just too perfectly evolved.
Exactly. To his mind, biological self-destruction was fundamentally impossible.
So Ehrlich essentially viewed the immune system as like a perfectly trained guard dog. It might bite intruders, it might even make a mistake and ignore an intruder, but it would absolutely never bite its owner.
That is a perfect analogy, and that assumption of absolute self-protection became the foundational dogma of immunology for decades. It was written into textbooks as an unbreakable law of nature. Yeah. The only problem is, while the guard dog can bite the owner, it just took over 50 years for the scientific community to prove it and correct the record.
Okay, let's dig into that moment, because shattering that unbreakable rule redefined the concept of auto or self forever. So fast forward to 1956 and 1957 at the University at Buffalo. We have an American pathologist named Ernst Witebski and his student Noel Rose.
Right. They were conducting experiments on rabbits trying to do exactly what Ehrlich said was impossible. They deliberately immunized these rabbits with extracts of the rabbit's very own thyroid tissue.
But wait, if they just injected the rabbit with its own natural thyroid tissue, wouldn't the immune system just ignore it? I mean, because it's self, how do they actually get the immune system to care?
That is the crucial mechanical step. They didn't just inject the tissue on its own. They mixed the rabbit's thyroid extract with an immune-boosting substance called an adjuvant.
Okay, an adjuvant, what does that do?
It's basically a cocktail of biochemical triggers, often containing killed bacteria or specific oils, that acts like a massive biological megaphone screaming danger to the immune system.
Ah, so they trick the targeting system.
Exactly.
The immune cells rush in because they hear the danger alarm from the adjuvant, but when they arrive, they see the rabbit's own thyroid tissue sitting right there next to the alarm. They get confused, associate the self-tissue with the danger signal, and just attack both.
And that biochemical trick worked beautifully. The rabbits developed severe targeted thyroid inflammation. Witebski and Rose had actively manufactured an immune attack on the self in a lab.
Which is incredible.
It was. It was strikingly similar to a human condition called Hashimoto's thyroiditis, which had actually been documented clinically back in 1912 but never fully understood until now.
Okay, let's unpack this.
Okay.
Because when we talk about the immune system attacking the body, a lot of people naturally assume that the immune system has just gone into overdrive.
Yeah.
Like it's a raw power issue.
Yeah, that it's too strong.
Right. But this isn't a case of the immune system being super strong.
Right. Not at all. That is a massive misconception. Autoimmunity is not about having too much immunity. It is fundamentally a regulatory failure. It's a problem of misdirection.
So it's like friendly fire in a military operation where the communication network has been jammed.
Yeah.
The soldiers aren't necessarily fighting harder, they just have the wrong coordinates.
Exactly. And the consequence of that jammed communication is that an autoimmune patient can have an immune system that is violently attacking their own healthy joint tissue or pancreas, while simultaneously leaving them completely vulnerable to a common cold or outside infection.
Because the targeting mechanism is just broken.
Precisely. Proving horror autotoxicus was wrong was monumental.
Because it opened the floodgates for understanding human autoimmune diseases as an entire category, right?
Yeah.
I mean, type 1 diabetes, where the immune system destroys pancreatic beta cells. Rheumatoid arthritis attacking joint tissue.
Multiple sclerosis attacking the myelin sheath of neurons. Lupus.
Yeah, they all finally made sense under this umbrella of autoimmunity. But I have to imagine that once Witebski and Rose proved the guard dog could bite the owner, the field of immunology was thrown into total chaos.
Oh, it triggered a massive philosophical and scientific debate. Because if the immune system is perfectly capable of attacking the self, how does it actually decide who the intruder is on a daily basis? What is the actual rule?
Right. If the ID badge scanner is broken, how does the bouncer know who to kick out of the club?
Well, for most of the 20th century, the dominant answer to that was Frank McFarlane Burnett's clonal selection theory, introduced in 1957. He argued that the immune system is fundamentally organized around self versus non-self discrimination. Basically, immune cells are trained very early in fetal development to memorize and ignore the body's own molecules and attack absolutely anything else that is foreign.
And this was backed up by Peter Medawar's famous skin graft experiments on mice, wasn't it? Which showed that tolerance to self-tissue is indeed a learned behavior during development. I mean, they even shared the 1960 Nobel Prize for this.
They did. But, and it's a big but, the self-non-self model has a really stubborn, glaring flaw when you look at how the human body actually operates in the real world.
Let me guess. If the rule is strictly ignore self, attack non-self, then we run into a huge problem with tumors.
Bingo.
Because a tumor is technically made of your own cells. It is genetically self, just slightly mutated. Yet a healthy immune system reliably spots and attacks tumors all the time.
That is the first major paradox. The second major paradox is the gut microbiome. You have trillions of foreign bacteria sitting in your gut right now.
And they are definitively non-self.
Exactly. If Burnett's rule was absolute, your immune system should be waging a constant violent war against your own digestive tract. But it harmlessly ignores them.
So the whole self-non-self binary is just too rigid. It doesn't explain the nuance. Who figured out the alternative?
In 1994, an American immunologist named Polly Matzinger proposed a genuine rival framework called the danger model.
The danger model.
Yeah, she argued that the immune system doesn't actually care about the philosophical question of is this self or non-self. Instead, it asks a much more practical question: is this tissue sending out damage or stress alarm signals?
Oh, wow. So Burnett's old model treats the immune system like a bouncer at a club, strictly checking IDs for self or non-self. But Matzinger's danger model treats it like a smoke detector.
That's right.
It doesn't care who you are or where you came from, it just cares if there's a fire.
And that perfectly solves the paradoxes. A tumor is self, but as it grows aggressively, it causes tissue damage and sends out distress signals. The smoke detector goes off, and the immune system attacks the self-tumor.
And meanwhile, a gut microbe is foreign, but it just chills there, helping digest food and causing no cellular damage. The smoke detector stays quiet, and the foreign bacteria are tolerated. This constant, messy negotiation, realizing that rigidly defended a pure self is actually a flawed biological strategy, is a perfect pivot back to genetics.
Oh, definitely.
Because, oddly enough, around this exact same time, genetics was discovering the exact same thing, that being genetically pure or same is often a massive disadvantage. Let's talk about the sickle cell trait and the power of being a heterozygote.
This is perhaps the most powerful example of relational biology we have. Let's look at the gene for hemoglobin, the protein in your red blood cells that carries oxygen. There is a normal version of this gene, which we call HbA, and a mutant version, HbS.
Going back to Bateson's roots, homozygous meaning same, heterozygous meaning different. If someone is homozygous for the mutant gene-like, they inherit 2 copies of HbS. They develop sickle cell disease.
And mechanically, what happens there is devastating. The hemoglobin molecules stick together and form these long, rigid fibers inside the red blood cell.
Oh, wow.
Yeah, and it distorts the cell from a nice, flexible donut shape into a sharp, rigid sickle shape. These rigid cells get stuck in tiny blood vessels, causing severe pain crises, progressive organ damage, and historically, a sharply shortened lifespan.
Right. And on the other extreme, if someone is homozygous for the normal gene 2 copies of HbA, they have entirely normal, flexible red blood cells.
Right.
But the biological magic happens in the middle. What if you are heterozygous? You inherit 1 normal HbA and 1 mutant HbS.
Then you have what's clinically called sickle cell trait. You
Yeah.
generally have healthy red blood cells and you don't suffer from sickle cell disease, but you get a critical life-saving bonus.
Right. In the early 1950s, a British geneticist named Anthony Allison was studying populations in East Africa, and he noticed something fascinating. People who were heterozygous carriers of the sickle trait were disproportionately common in regions where malaria was endemic.
The environment was actively selecting for the mismatched genes.
But how does that actually work mechanically? Like, how does a mismatched blood cell fight off a mosquito-borne parasite?
The mechanism is incredible. When the malaria parasite Plasmodium falciparum infects a normal red blood cell, it consumes the cell's resources and drops the oxygen level. In a normal homozygous HbA person, the parasite just thrives and multiplies.
But in a heterozygous person,
That drop in oxygen triggers a unique reaction. It causes only that specific infected cell to collapse into a sickle shape.
wow, so the cell essentially self-destructs to flag down the immune system.
Actually, it flags down the spleen.
Oh, the spleen?
Yes, the spleen acts as a blood filter. When the infected heterozygous red blood cell sickles, the spleen immediately recognizes it as damaged, pulls it out of circulation, and destroys it, taking the malaria parasite down with it. The heterozygous cells basically use the mutation as a targeted weapon.
That is wild. And later epidemiological studies, including those massive ones published in The Lancet in the early 2000s, they confirmed this completely, didn't they? Children carrying the trait experience substantially lower rates of severe and cerebral malaria.
They absolutely do. It's a phenomenon evolutionary biologists call balancing selection. The heterozygote is biologically fitter than either of the homozygote extremes.
Here's where it gets really interesting, because it completely flips our usual expectations of genetics. We often think of biology as seeking perfection or uniformity. We assume 2 normal, matching genes must be the best possible outcome.
But in an environment with malaria, being different is actually better for survival than either extreme of same.
And it debunks a really common misunderstanding about that hetero prefix in genetics. People hear heterozygote and assume it just means a symptomless carrier of a recessive disease.
Right, but it literally just describes the mismatch. The outcome of that mismatch varies wildly. Sometimes the normal gene completely masks the mutant one. Sometimes you get incomplete dominance resulting in a blended physical trait.
Or sometimes both traits show up simultaneously, like in AB blood types, where you express both A and B antigens.
Exactly. And sometimes it gives you a unique evolutionary advantage against a deadly parasite.
It is a profound reminder of how much precision these Greek roots brought to our understanding of the body. But it is equally important to look at how this exact terminology left the laboratory and was historically misused, because that same veneer of scientific precision can be deadly when the terminology is weaponized.
That brings us to a very dark chapter in the early 20th century. The American eugenics movement essentially hijacked William Bateson's careful terminology of homozygotes and heterozygotes.
Yeah, they took the vocabulary Mendelian ratios, which accurately describe simple, single-gene traits like pea plant colors, and they aggressively applied them to incredibly complex human social conditions.
They falsely claim that things like poverty, criminality, or what they broadly categorized as feeble-mindedness were controlled by single genes in a simple, dominant, and recessive fashion.
Which is biologically absurd.
Yeah.
I mean, human behavior, intelligence, and social outcomes are not dictated by a single recessive pea plant gene. They are the result of massively complex interactions between thousands of genes and the environment.
It was completely bankrupt on its own scientific terms, but because eugenicists wrapped their social prejudices in this precise-sounding genetic terminology, using the language of same and different alleles, it gave their policies a false authority.
It's horrifying. This pseudoscience directly underpinned forced sterilization laws across dozens of U.S. states. These were laws that were shockingly upheld by the U.S. Supreme Court in the 1927 case Buck v. Bell.
It is a stark historical warning about what happens when the vocabulary of biology is used to dress up bad science and enforce a social agenda.
Absolutely.
Hmm.
But it wasn't just social policies where the language of same and different caused dangerous confusion. The medical field itself actually had to physically retire certain words because the shifting definitions were causing lethal misunderstandings in surgery.
The best example of this is transplant nomenclature. For most of the 20th century, if a surgeon moved a kidney between two unrelated humans, they called it a homograft.
They were using homo for same because the organ was moving between two members of the same species, human to human.
Right. But as our understanding of genetics deepened, that word collided head-on with terms like homozygous. Think about it. A kidney from an unrelated donor is emphatically not genetically identical to the recipient.
So calling a genetically mismatched kidney a homograft was etymologically dangerous.
Very dangerous. It implied a genetic sameness that didn't exist. It was a graft between two totally different genomes.
So they had to change the word to save the meaning and prevent clinical confusion.
By the 1970s, international medical committees stepped in and officially replaced homograft with allograft, using the Greek root allos, meaning other.
But scientific literature has a long memory, right? If you read pre-1970s medical journals, you'll still see homograft rejection describing what we now call allograft rejection.
The shifting ground of vocabulary is fascinating. And while we are clearing up etymological confusion, we absolutely have to mention the ultimate false friend here, Homo sapiens.
Oh, yes. It is the most common mix-up in biology classes.
It really is. The Homo in Homo sapiens is Latin. It means man or human. It has absolutely zero relation to the Greek prefix homo, meaning same.
They have the exact same spelling, but completely unrelated origins and meanings.
Exactly.
Okay, so we've seen how same can be confusing, both historically and linguistically. Let's look at how different is completely upending modern medicine right now, specifically in cancer treatment. There's a landmark study in 2012 by Marco Gerlinger and his colleagues that redefined how we view tumors.
Published in the New England Journal of Medicine, they were looking at renal cell carcinoma, a type of kidney tumor. Traditionally, a biopsy involves taking one sample from a tumor to see what mutations are driving it. But Gerlinger's team did something different.
They sequenced the DNA from multiple physically separate regions of the exact same tumor mass in several patients, right?
And the results were staggering. The different regions of the same tumor did not share the same mutations.
They found that different parts of a single tumor had actually branched off genetically from each other.
Right. They were evolving independently, adapting to local conditions within the same mass. The field now calls this intertumor heterogeneity.
And this isn't just an academic curiosity. This explains why cancer is so hard to cure. Because if an oncologist assumes a tumor is homogeneous, like uniform all the way through, they might use one single biopsy needle, find a specific mutation, and prescribe a highly targeted drug that works perfectly on that one spot.
But because of intratumor heterogeneity, they are completely missing a drug-resistant genetic dialect hiding on the exact opposite side of the tumor.
Wow.
Yeah, so the cancer looks like it's responding beautifully to the treatment, the initial mass shrinks,
No.
but then it violently relapses from a subpopulation of cells that the targeted therapy never even touched.
So a tumor isn't just one self. It is a heterogeneous community of others, all living under one roof.
That's exactly it.
So what does this all mean? We've traveled from Mendel's pea plants and Ehrlich's guard dogs to malaria resistance and cancer biopsies. Why does understanding the precise mechanics of auto, hetero, and homo matter for you listening right now?
It matters because modern medicine relies entirely on navigating these boundaries. When doctors treat aggressive blood cancers, they vastly prefer autologous stem cell transplants, harvesting and returning the patient's own cells, precisely because self-tissue avoids graft versus host disease.
Right. There's no risk of the transplanted immune cells waking up, recognizing the patient's body as other, and attacking it.
Which is a major life-threatening complication in allogeneic or foreign transplants.
This vocabulary also guides genetic counseling for parents trying to understand their heterozygous carrier status for conditions like cystic fibrosis, right?
Absolutely. And these prefixes literally built the everyday vocabulary you use to describe the human experience. When the Swiss psychiatrist Eugen Bleuler coined the term autism in 1910, he used autos, meaning self, to describe what he viewed as a pathological self-absorption in patients.
A term Leo Kanner later repurposed into the modern diagnosis in 1943.
Yes, and words like homosexual and heterosexual. Neither of those existed until the late 1860s when a writer named Carl Maria Kurt Benny coined them by hybridizing these exact same ancient Greek prefixes with Latin suffixes. They truly are the skeleton key to understanding how we categorize life.
They really are. And I want to leave you with one final thought to ponder, bringing us all the way back to that opening question about relational identity. Think about biological chimeras.
Oh, this is a great concept.
Right. These are organisms, including humans, that contain cells from two genetically distinct origins in one single body. For example, fraternal twins who harmlessly exchange stem cells with each other while still in the womb.
They are born, and they walk around for their entire lives with two different sets of DNA in their bloodstream.
They are a living, breathing blur of the self versus other line, and they are perfectly healthy. So if nature already effortlessly creates chimeras, organisms that don't violently attack the other inside them, will the future of medicine eventually move away from trying to violently defend a strict boundary of self?
It's a profound question.
Could the ultimate cure for autoimmune disease or transplant rejection be teaching our immune systems to gracefully accept being a mosaic of others? Something to think about. Thanks for taking this deep dive with us.